Hello everybody, in this video lecture I will cover chapter 9 part 1 DNA structure and replication. We're gonna begin with DNA timeline. So if you look at this diagram here, you can see Mendel's experiment in 1865, and Mendel didn't know much about DNA or chromosomes because chromosomes and DNA then was observed and described a little bit later than his time. We will briefly talk about Griffiths. What Griffiths did, Griffiths pretty much proved that inside a cell there is some molecule, right? Or there are some molecules, and those molecules carry instructions for cells how to function. So they have instruction on morphology and physiology of the cell. And these molecules, Griffiths didn't know what kind of molecule it was. He didn't know if it was DNA or proteins or lipids or carbohydrates, but he proved that molecule like this exists, and this molecule can be passed from one cell to another cell, from generation to generation, and that molecule has instruction about cell functioning. So it took many other experiments over here, Avery, McCarthy, Franklin, actually Franklin more to this point, but Hershey and Chase. And what this experiment proved, they proved that that molecule that Griffiths described in his experiments, that molecule was DNA. And then Watson and Crick, they discover structure of DNA and they get Nobel Prize for their work. And you can see how our discovery continues from 1960s, 1980s to today's day, and we're still learning more and more information about DNA. So Mendel's experiment proved that molecules from the parents were transferred to offspring. But what type of molecule? Could it be a lipid, protein, nucleic acid, carbohydrates? Scientists narrowed it down to two possibilities, nucleic acid and proteins. And I just want to remind you that nucleic acids are DNA, right? And a smaller molecule is RNA. And we know that proteins are polymers of amino acid. And why scientists down it to two possibilities? Because those are the most complex. Out of these four, lipids and carbohydrates, they're not very complex molecules, have pretty simple structure. DNA and proteins are complex. So that suit better to be this molecule of inheritance. So on this slide, we will briefly talk about Griffith's experiment. So what Griffith did, he had a bacteria that caused pneumonia, and bacteria has two different strains. So one strain is called R bacteria and another is S bacteria. So when you inject mice with R bacteria, right, mouse remains healthy. So the conclusion would be R strain does not cause pneumonia. When Griffiths injected mice with S strand, right, mouse died. And conclusion was S strand caused pneumonia. So very, reasonable conclusion that makes lots of sense. You inject with R strand, mouse lives, so R strand doesn't cause pneumonia. Inject with S strand, mouse dies, S strand causes pneumonia. But now what Griffiths did, he takes this S strand and he heats it. He heated it and he kills S bacteria. Inject a mouse, mouse remains healthy. Conclusion is heat killed, strain doesn't cause pneumonia. But that was interesting about Griffith's experiment, step number 4. When Griffith takes this R strand and he takes this one, heat killed S strand. Neither one of them caused pneumonia. Now he mixes them, inject the mouse, and mouse dies. Right, so when they are on its own, when he used them separately by itself. R strand doesn't cause pneumonia. Heat killed S strand does not cause pneumonia. But when he mixed them together, something happened inside this mixture, right? Something that allowed this bacteria to kill the mouse. So think about this way, and this is what Griffith thought. So here's our blue, that's our R strand. So Griffith's hypothesis was there is some molecule inside R strand. And that molecule that has instruction for this bacteria to divide, to get its energy, to perform all its function, physiological function, including, not causing pneumonia, right? This bacteria lives, it reproduces, but it does not cause pneumonia. This S strand also has this, molecule. And this molecule has instruction on how to divide, how to obtain energy. And here's a little piece of this molecule that has instructions how to cause pneumonia. So when Griffiths killed this S strand, so you're killing the bacteria, but what he didn't destroy, this molecule. And this molecule was somehow transferred to R strand. So here we have this molecule, right, with this gene that has instructions. Here's the instruction how to cause pneumonia. And it's transformed R bacteria into S bacteria. So Griffiths didn't know what molecule it was. We know today it was DNA. So here's his experiment. Inject mice with live R and mice live. Inject with S, mice die. Inject with dead S, mice live. But then when we mix live R and dead S, you would predict that it should not kill the mice, but mice dies. Right? So here was interpretation of his results. Heat kills S cells, but not hereditary material. This material was transferred to R cells, transforming them into S cells through the process called transformation. And it was permanent change. After 100 of generation, transformed R cells still contain the instruction of causing infection. But Griffiths couldn't find out in his experiment what kind of bacteria it was. But Avery did. So what Avery did, so the same mixture, DNA, RNA, and proteins, right? So here's the bacteria. And bacteria doesn't only have a DNA. So it has here the DNA, it has RNA, and some proteins, right? It's all three molecules. So we kill the bacteria and we have these three molecules in the mixture. We put it into the syringe, right? Here's one syringe, here's another syringe, and here's another syringe, right? But what Avery added, in one syringe, he put protease, right? That's a protease. And what protease does, it destroys protein. So no protein in this syringe. In this syringe, no RNA. And in this syringe, no DNA. So in all of them, one molecule was destroyed. And what he noticed that only if you have DNA, the transformation happens. You don't have DNA, no transformation. That means that transformation only occurs when DNA was present. DNA must instruct the cells to switch from R phenotype to S phenotype. So here we have interpretation of Avery results. If add protein digesting enzymes, protein destroyed, but DNA intact. So DNA intact, then we have transformation into S cells. If DNA destroyed, then no transformation. Here you can see another experiment, Hershey and Chase, and they used not bacteria this time, but bacteriophage. And bacteriophage is a virus of bacteria. And as a virus, it has, simple structure. It has only DNA and proteins. So it was easy to prove that DNA was the molecule of inheritance. So Hershey and Chase experiment proved beyond any doubt that DNA is a molecule of inheritance. But what did it look like? To answer this question, a scientist needed the work of Chargaff's first. Chargaff's law pretty much told us that in a DNA we have 4 different nucleotides, right? A, T, C, and G. And Chargaff's law says that amount of A and T is almost the same, and amount of G and C is the same. That means that they are paired in some way. That means that A is somehow paired with T and C paired with G. Another piece of evidence that allowed scientists to discover structure of DNA was the work of Franklin. And Franklin, she took picture of DNA using X-ray, and every time she get a picture, she saw this cross, but she couldn't figure out structure based on her photographs. She couldn't, but these 2 scientists, Watson and Crick, they did. So they using the previous data from all scientists and experiments. They published 1-page paper on solving the structure of DNA. So here they build the structure of DNA, and they get a Nobel Prize in 1962. So what is the structure of DNA? DNA is a nucleic acid. It's polymer made of monomers, and monomers of DNA are nucleotides. And you can see picture over here of nucleotide, or I can draw it for you. So each nucleotide has a sugar, then phosphate base, and phosphorus, right? Phosphate group. So phosphate group, sugar. So here sugar is specific. It's deoxyribonucleic acid. So I put D over here. And DNA has 4 different nucleotides. So it can be adenine, then we put A over here. Or it can be thymine, and I put T over here. Or it can be cytosine, I put C over here. Or it can be guanine. So DNA is made of nucleotides, and nucleotides are made of deoxyribose, that's a sugar, phosphate group, and nitrogenous base. DNA is also double helix, so you see over here the double helix, and here's our nucleotides. Right, so here's a sugar, phosphate group, and the base. And sugar and phosphate, they form sugar-phosphate backbone. So I already told you that we have 4 different nucleotides, T, C, A, and G, or thymine, cytosine, adenine, and guanine. And DNA is double helix. Double means it has 2 strands, and helix means a spiral. And here's our base pairing rule. Adenine always with thymine, and cytosine always with guanine, and we call it complementary bases. RNA structure. Like DNA, RNA is polymer of nucleotide, and it's also made from sugar, phosphate group, and nitrogenous base. But sugar is different. It's not deoxyribose, it's actually ribose. Same phosphate group and different nucleotides. So RNA does has adenine and cytosine and guanine, but it doesn't have T. So instead of thymine, RNA has uracil. So you can see the different differences in structure. Here's the DNA, double helix with the thymine, and this is single-stranded RNA, and it has uracil instead of thymine. Now DNA must replicate itself. DNA must replicate itself so cells can pass their DNA to a new generation. So when a cell divides, it's important that each daughter cell receives identical copy of the DNA, and it's accomplished by the process of DNA replication. DNA replication occurs during S phase of a cell cycle. So that's a cell cycle, and cell cycle, it's like a pie diagram. So one part, that M phase, this is where a cell divides, so it's mitotic phase. And interphase is divided into G1, S, and G2, and that's our S phase of interphase when DNA replicates. So it's interphase, and this is S phase of interphase. DNA replication is called semi-conservative. And semi-conservative means that only part of the original DNA is conserved during replication. So look at this picture here. This is parental DNA, parental molecule, and it's shown here in blue color. So both strands are blue. When we're looking at daughter DNAs, and here's our daughter DNAs, one strand is blue and one strand is red. Well, of course, inside the cell, we don't have blue and red color. This is just to help you to understand that new daughter DNA is half new and half old. So one DNA molecule, after replication, became two DNA molecules. Each consists of a new and old strand. And a new strand is made using old strand as a template. So here's a question asking you, what would be the complementary strand for the following DNA sequence? So if I have DNA over here, right, so I'm gonna just kind of like open it up. So here. And if this strand has nucleotides, so I'm gonna use this ACG, so it has A, C, G, T, and A. So the question asking you, what would be this second strand, complementary strand? And all you need to do, use a base pairing rule. A always pairs with T, so that's gonna be T. This is gonna be G, C, A, and T. And the second question asking you, well, what would be the sequence of the replicated DNA of daughter strand? So again, so here's the DNA molecule. Right, so I'm gonna again unzip it like this. And if one strand has this DNA sequence, and I'm just gonna put some of them like T, T, A, and C. So here's our daughter strand that will be synthesized based on this Chargaff's rule or base pairing rule. So this will be A, T, and G, and so on. We do have answers to these questions on our next slide. Okay, so here are the answers. You can pause the video and check those answers. Now, DNA replication begins with a replication fork, right? And it's kind of like we have this enzyme called helicase, and helicase unwind and uncoil DNA breaking these hydrogen bonds between bases, nitrogenous bases. And then this enzyme called DNA polymerase. It's DNA polymerase, and this is DNA polymerase. It's gonna synthesize, DNA polymerase synthesize this new strand or daughter strand of DNA over here and over here. One DNA strand is called leading strand, and DNA synthesized continuously over here. So look, that's a continuous synthesis. And another strand is called lagging strand. That's a lagging strand. And DNA is synthesized in the fragments. So here we have some fragments, and those fragments are called Okazaki fragments. And then another enzyme, DNA ligase, joins these fragments together. That was the last slide, and I hope it was helpful.